So in the second section of Chapter 9, again, continuing to look at microbial genetics, in this section we're going to focus on protein synthesis. And so, again, if you recall from biology that in order to make proteins, it all begins with DNA, right? But it's not just solely based on DNA. If you remember, in protein synthesis, you start with DNA and you transcribe it or condense it into a smaller version known as RNA. And so remember, RNA is similar to DNA in that it is a nucleic acid. It's made of phosphates, sugars, and bases, but there are a few differences that we see. The first is that RNA is single-stranded. So where DNA is a double helix, RNA is half of the size, so it's much smaller, which for eukaryotes, if you recall, is beneficial because it can actually fit through the nuclear pores, leave the nucleus, and then make its way to the ribosome, where it can actually be used to help make proteins. The other thing we know about RNA is, yes, it also has 4 bases, but the bases are slightly different. In RNA, it still has adenine, guanine, cytosine, but there is no thymine. Instead of thymine, it's replaced with the base uracil. And then again, RNA is also made of sugars, but the sugar is different. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. The other thing that we note about RNA is that there actually are 3 types of RNA. Again, if you remember from biology, you have messenger RNA, transfer RNA, and then ribosomal RNA. And so again, just really quickly, if we look at these, so the messenger RNA, it is the message. It's what contains the instructions for the amino acids that are needed, the sequence that are needed in, so that you can build the appropriate protein. And so typically what messenger RNA does is it goes from the DNA to the ribosome. So it carries that message, carries those instructions, telling the ribosome which proteins to build. Transfer RNA, the way that I describe it in biology, is similar to the Lowe's delivery truck. It's bringing the materials to the site so you can build what you need to build. So in this case, transfer RNA is bringing the amino acids to the ribosome in the correct order so that a protein can be made. And then the third type of RNA is ribosomal RNA. Its job is basically to help build ribosomes that are going to be used in protein synthesis. So again, here's your basic RNA. So again, there's your messenger RNA carrying those instructions. There's your phosphate sugar, right? Phosphate, sugar, and then base that's sticking out from it, right? And so this is carrying the instructions. They're going to be used to make proteins. Again, there's your transfer RNA. Transfer RNA is going to bring the appropriate amino acid to the location where it's needed. And then just kind of reiterate from biology the process of protein synthesis. So if you look at objectives 7 and 8, they talk through transcription and translation. So just a quick reminder, transcription is where you take the DNA and you use it to make RNA. So in this case, you take the double helix, you're going to uncoil it, just kind of like you did with replication. You split it in half, and then you use that DNA as a template to make RNA. So the biggest structural difference we'll see here is instead of adenine binding with thymine, since you're making RNA, adenine is going to bind with uracil, right? And so transcription ends with the new RNA sequence that was just made from DNA breaking away from those DNA bases and then moving towards the ribosome. And then in translation, that RNA is then used to make the protein. And so this is where we see the transfer RNA bring those appropriate amino acids to the location so that they can then do what's needed. Okay, so just a quick reminder of that process, and we'll go back over this in class because I know for some of you it's been a while since you had biology. Some of you may have just cleared the test, and so a lot of this stuff may sound new to you, and so we will take some time to go back through this process of taking the DNA, using it to make RNA, that's the transcription part, and then using that RNA to build an amino acid sequence or a protein, that's the translation part. Now, I'm going to scoot through to here because Objective 8 says that there are some differences between prokaryotes and eukaryotes in transcription translation. And so, biggest differences, we're going to see a couple things that are listed here. First off, the start codon. Remember, the start codon is AUG. It tells us where to begin reading the sequence. And so, for prokaryotes and eukaryotes, it's the same start codon, but in prokaryotes, it stands for a different form of an amino acid methionine. Other things we see, of course, the nucleus in eukaryotes keeps transcription and translation from happening at the same time. So in eukaryotes, transcription has to happen first, and then it's followed by translation. In prokaryotes, because there's no nucleus separating the two processes, they can be happening simultaneously. So that's kind of a difference that we see there. Some other things they mention in eukaryotes. So eukaryotes are not one uninterrupted series of codes. It has both introns and exons that are kind of separating those active units. Prokaryotes, we don't see that. So in prokaryotes, it's just one long interrupted sequence, and so there is no splicing that has to occur. And again, we'll go back over this in more detail in lecture, because I think in lecture I can give you some really good examples to help you note differences here. But again, this is what it looks like in a eukaryote. So you've got exons and introns. Introns are in the way, right? So before they can actually finish protein synthesis, these have to be removed or spliced away, and then the exons become an active strand of RNA. You don't see this in a prokaryote, and so that's kind of a difference that's there. Okay?